as concerned the fermented design modules, this module will deal with the cleaning, sterilization and the sampling facilities. We know that if we have a fermenter that have to be used for number of operations and for a continuous use. So just one batch to another batch, we need the cleaning. So the small scale reactors are taken apart and then cleaned before being reassembled. And then mostly their cleaning is carried out ex situ. So we just disassemble the whole fermenter and then we utilize them for autoclave. And then in other case, when there is a large fermenters, then we have to clean them in situ. That is known as clean in place, CIP. So the CIP involves the complete cleaning of not only the fermenter but also all the line, sump and dump line system, all the internal component of the system, acid addition system, alkali delivery system, aeration delivery system. So there's all the system should be properly cleaned. So that's why the cleaning, and then after the cleaning and before running the fermentation process, the sterilization of all the line is very critical. So that's why that should be the provision of that. So in this slide you can see that there's a different lines. Mostly as concerned the sterilization, that is mostly in a large scale fermenters happen through the steam. So all the have the control, all that is controlled by the different valves, automatic valves, and then the supply from, you can see here in this slide that the blue color show the inside of the fermenter. So and these are, the red line show the steam that having the different valve C, valve A and valve B in this case, in this case and in this case. So if the pump A will on, then that steam can pass into into the fermenter, but that is only needed when we have to clean, either to sterilize the fermenter inside. But sometimes this fermenter insides can only be supplied the jacket, not inside the fermenter, just to raise the temperature. But in case of another, you can see that sometimes the material and others. So this is only happen when we say that in case of the cleaning, but sometimes we have to check the quality of the product inside. So the sampling provision should be in a fermenters. We have to take some samples. So in some case there is a different valves and from where we can get the samples, and at the time of the sampling then there should be a proper sterilization. Otherwise when at the time of the sample collection, then there is a risk of contamination. So that's why we should be very critical about in case of that. So then we can see that by this whole process we can use the different line system. So in designing the fermenter, we should be care about all such kind of the provisions for a fermenter.